guys, welcome back to Swaggy Science students.
Aaron's feeling a bit under the weather.
A little bit. This is chapter 17, Organizing Life's Diversity. Yay. So our first vocab word is classification, grouping organisms based on a set of criteria. So Aristotle was the first person to come up with the first system of biological classification. It wasn't really very complete. It had some loopholes. So the first thing was like plant or animal. Second thing, if it was does it have blood, type of habits, behaviors, body shape, all that good stuff. Step 3, if the organism is a plant, what the plant size is, plant structure, was it a tree, a shrub, an herb, and when you miss, the next vocab word is taxonomy, in identifying and naming and clarifying species. So it's more specification. Next we got binomial monoculture. So each species has 2 scientific names. So the first part is the genus name. The second part is the species specific name. The binomial monoculture is based in Latin because Latin is a dead language and it doesn't change. It's officially spoken. It's not officially spoken anymore. That's why it's dead, which means it doesn't change. Part of the so we're going to talk about the taxonomic categories. Part of the hierarchical system, each category contained within another. So it's arranged from broad to very specific. Taxon comes from the word taxa, which means a group or organisms. Genus, which comes from the Latin term genera, a group of species that's closely related and shares a common ancestor. And then family contains related like genes. Order contains related families. Class contains related orders. Phylum comes from the Latin word phyla, contains related classes. Kingdom contains related phyla, and domain contains one or more kingdoms.
So now we're going to be talking about modern classification. So determining species. First, the typological species concept, which each species is different based on physical similarities, or judging a book by its cover. Biological species concept is organisms that interbreed and produce fertile offspring. Phylogenetic species concept, the word phylogeny is the evolutionary history of species, so this would be species that are a distinct cluster of organisms. Next we're going to be talking about characters. Characters are inherited features that vary among species. The morphologic characters, the denotes share characteristics. So that species that share morphologic characters are closely related and have a recent common ancestor. Biochemical characters determines evolutionary relationships. The more recently those groups share a common ancestor.
So now we'll be talking about domains and kingdoms. So the grouping of species, modern biological classification and taxonomy system has 6 kingdoms and 3 domains. So the domain bacteria, very diverse, can survive in many types of environments. Domain archaea, ancient bacteria with diverse nutritional requirements and live in harsh environments. Domain eukaryotes, everything that is not a bacteria and has a nucleus in all cells. So the kingdom Protista, protists are unicellular, multicellular, and live in colonies. Protists can be plant-like, animal-like, or fungus-like. Kingdom fungi Unicellular or multicellular, have cell wall, absorb nutrients from organic materials and environments. Kingdom plantae, multicellular and have cell walls composed of cellulose. Kingdom Animalia, all are heterotrophic, multicellular eukaryotes, and has a complex organ system. Now we'll be talking about Chapter 18.
Chapter 18, Viruses and Bacteria. So first we're going to be talking about bacteria. First is the diversity of prokaryotes. Bacteria is a microscopic prokaryotic organisms. So first, eubacteria. Cell walls of this bacteria are made of peptolyncan, a protein, and a carbohydrate. The next is the archaebacteria, and that's hot, acidic environments, salty environments, and they give off methane in no oxygen environment. So next we'll be talking about structure of bacteria. 3 vocab words. Nucleoid, that contains circular chromosomes. A capsule is polysaccharide layer, polysaccharide, layer on the cell wall. Pili is hair-like structures, and they help bacteria attach to surfaces. So now we're going to be identification of bacteria. So first you have to know the shape. 2, the movement. A flagella is like a whip-like tail used for locomotion, so that could be movement. Next, we're going to be talking about reproduction of bacteria. So, binary fission is when one bacterial cell divides into 2 genetically identical cells. Conjugation is 2 bacteria cells exchanging information. Woohoo!
Yeeter. Now we'll be talking about metabolism and prokaryotes. So photoautotrophs
that contains circular chromosomes. A capsule is polysaccharide layer, polysaccharide, layer on the cell wall. Pili is hair-like structures, and they help bacteria attach to surfaces. So now we're going to be identification of bacteria. So first you have to know the shape. 2, the movement. A flagella is like a whip-like tail used for locomotion, so that could be movement. Next, we're going to be talking about reproduction of bacteria. So, binary fission is when one bacterial cell divides into 2 genetically identical cells. Conjugation is 2 bacteria cells exchanging information. Woohoo!
Yeeter. Now we'll be talking about metabolism and prokaryotes. So photoautotrophs do photosynthesis. Chemoautotrophs do chemosynthesis. Aerobes use oxygen to break down ATP. Aerobes do not use oxygen to break down ATP. Survival of bacteria. So they're endospores. To survive extreme heat and cold, dehydration, and UV rays, some bacteria makes an endospore to protect themselves until conditions become more suitable. Then there are mutations, short lifespan of bacteria, and high reproductive rates. With high mutation rates and mutations give genetic diversity, which is pretty cool, I guess. Nutrient cycling and nutrient fixation. So bacteria are decomposers and detrivores. They're nature's recyclers. Pretty cool. Normal flora, harmless bacteria. Yeah, pretty much. Normal flora. Most bacteria live on us and in us, which is kind of gross. Kind of not it. So yeah, get out of me. Food and medicine. Bacteria aided for processing foods, so like yogurt and antibiotics. Yeah. Disease causing bacteria, so there's a small percent, less than 1% of bacteria actually cause diseases, which is pretty weird because why am I having the 1% at the moment? These disease-causing bacteria multiply quickly and infect their carrier, me. So now we'll be talking about viruses. Viruses are non-living strands in code. So like yesterday I got my flu shot and they put a strand of virus in me. So that's kind of not about it.
Not having it.
Not having it. Viruses have no organelles. Viruses can't make proteins move or replicate on its own. So the size, it's about 5 to 300 nanometers, and 1 nanometer is a billionth of a meter. So that's a pretty big number. Pretty dope.
So the origin of viruses most likely came from parts of cells. So they have similar to cellular genes. The structure, a capsid is the outer layer of a virus, which is made of protein. Viruses' genetic material is DNA or RNA, never both. Next, we're going to be talking about infection. In order to replicate, a virus must enter a host cell. The virus attaches to specific receptors on the plasma membrane. Once the virus is attached, genetic material inserted or stuck into the host cell. So, lytic cycle is a reproductive cycle. The virus tricks host cells to making many copies of viral RNA or DNA. The lysogenic cell is viral DNA becomes permanent DNA of a host cell. Yikes. Now we're going to be talking about retroviruses. An example is an HIV. A retrovirus is viruses that have RNA instead of DNA for genetic material. Next we're going to be talking about prions. Example is mad cow. Prions is a protein that can cause infection or disease, cause normal proteins to mutate or have a mutation. And
So yeah, that's pretty much it for chapter 17 and
And
chapter
18.
18. And my eyes hurt right now, so bye.
that's how you know it's a bad day.